Hello. I'm going to be talking to you today about my fourth year project, which I completed in Dr. O'Malley's lab here in UCC. And I was looking at how L cells are key to cross-barrier signalling to the host peripheral nervous system by GLP-1 secreting probiotics. So we were looking at the gut-brain axis, this bidirectional communication axis between the gut and the brain, which we know is important for regulation of homeostasis, primarily through regulation of gut functioning, so more permeability, absorption, secretion, energy balance. And it is believed that it signals one signals to the other using endocrine, neural and immune signalling. However, the exact mechanisms are largely unclear. This gut-brain axis is now, however, being extended to the microbiome gut-brain axis because we now see that the alterations in the gut microbiome composition, so microbial dysbiosis, has been shown to impact on the functioning of this gut-brain axis. Alterations can occur for several reasons, more predominantly the genetics, but also diet and antibiotic treatment, and also birth-related differences, for example, mode of delivery. These alterations that occur in the microbial composition have been shown to be associated with several disorders, so metabolic disorders such as obesity, but also gastrointestinal disorders and central nervous system disorders. However, to understand how an alteration in microbial composition can actually impact on the functioning of the gut-brain axis, we need to understand how this axis is working in health conditions. So the exact cellular and molecular mechanisms were our focus for this project. And we do know, however, that the vagus nerve is a key player in this, as shown by Bravo et al. And they cut the vagus nerve in a mouse that probiotic treatment, which showed neurochemical and behavioural changes in healthy mice, were not observed in the mice in which the vagus had been cut. In order to signal to the vagus nerve, the luminal contents must be sensed by the epithelium. So we believe that the L cells are potentially sensing the luminal microbial moieties. So these L cells are found embedded throughout the gut epithelium, and they are found to increase in abundance down the gastrointestinal tract, with highest abundance potentially in the colon, also where we find the highest abundance of the gut microbiota. So these L cells normally work to secrete GLP-1. They sense the nutrients in the apical membrane, and GLP-1 is secreted through the basolateral membrane. GLP-1 is this incretin hormone and works to decrease blood glucose through release of insulin and glucagon, but it can also modulate gastrointestinal function. So Dr. O'Malley's lab have recently shown that they can actually stimulate the myenteric neurons and increase colonic motility. What we do know is that the L cells can actually sense microbial derived factors, such as indole. So this signals to us that it could be potentially involved in this microbiome gut-brain axis signalling. The anatomy of the L cell also suggests it to be a sensor, as they have long basal processes termed neuropods with pre- and post-synaptic proteins, and they can synapse with neighbouring cells, for example, innervating nerve fibres. So GLP-1 could potentially be secreted from these L cells and stimulating vagal motor afferents, innervating the distal colon. And the expression of GLP-1 receptors in the dorsal root ganglion cells suggests that could potentially be occurring. So the aim of this project was to determine if a particular probiotic strain, so Lactobacillus paracasei, which has been genetically modified to secrete GLP-1, if it's stimulating rat colonic, intrinsic and extrinsic neurons, thereby potentially showing us a cellular or molecular mechanism underlying the relationship between luminal microbiota and the host peripheral nervous system. So we used a number of methods to investigate this, and we primarily looked at local signalling, so the submucosal neurons just underlying the mucosa. So we initially used immunofluorescence. So we took sections of the distal colon of Sprague-Dawley rats, as they provided an intact epithelium for us to investigate, and we stimulated them with several of our controls and our bacterial secretions. So these are, which we term supernatants. So these are the secretions, not the probiotics themselves. And we also stimulated them with the washed bacteria. So these are the probiotics. We then removed the mucosa from our sections so that we could expose the submucosal neurons and fix them so that we could look at them. We then fixed and blocked them and permeabilized them, and we labeled them with our primary antibodies, so anti-GLP-1 receptor and anti-CFOS. We then used calcium imaging to also look at this local signalling. So we took sections of our colon, but we removed half the mucosa to expose the submucosal neurons so that we could apply our controls and our supernatants and our bacteria, or just our supernatants and controls, to the mucosa but required from the submucosal neurons. And we loaded these with our calcium indicator fluo 4AM. So looking at the results of the immunofluorescence, initially we see that there is a high cytosolic expression of the GLP-1 receptor in these submucosal neurons, and this is largest in response to the supernatants, so the secretions from the genetically modified probiotics. And then looking at CFOS, so this is our marker for submucosal activation, we see that it is largely nuclear in response to the supernatants, suggesting to us that these GLP-1 secreting these GLP-1 receptor expressing neurons are responding to the probiotics indicated by our nuclear expression of CFOS. Moving on to our calcium imaging, we see a similar result here again, that the supernatants, so these are the secretions from the probiotics, are producing the largest stimulation of these submucosal neurons. But in addition to the immunofluorescence, what we see here is after applying a GLP-1 receptor antagonist, by reapplication of the supernatants, the response was significantly dampened, suggesting to us that it's the GLP-1, which we know are in the supernatants, that is responsible for this stimulation of the submucosal neurons. Moving on then to more extrinsic signalling, we used a method which has been developed by Dr. Maria Buckley here in UCC, and what we did was we were looking for vagal afferent stimulation. So taking a section of our distal colon with the mesenteric and vagal nerve intact, we then placed this into a nerve gut bath. So this consists of two chambers, as we can see down here on the right, two chambers. On one side we place the tissue, and on the other side we have the nerves, and this is filled with Krebs saline solution. So by putting the tissue on in one chamber, we can apply to the mucosa and ensure that we are recording from the nerve with our platinum bipolar electrode, and we applied our several controls, our supernatants and our washed probiotics. Looking at the results of this then, we see that again, similar to the immunofluorescence and the calcium imaging, it is the supernatants that are producing the largest response and the largest stimulation of these afferent vagal nerves innervating the distal colon. And again, when we apply the GLP-1 receptor antagonist, the response to reapplication of the supernatants was significantly dampened, suggesting it is the GLP-1 and the supernatants that is responsible for this stimulation. So bringing all this together, what we found is that these probiotics genetically modified to secrete GLP-1 are found to increase CFOS expression in GLP-1 expressing submucosal neurons. However, the probiotics themselves did not change the CFOS expression, so it is their secretions that are responsible for the stimulation. The bacteria, similarly with the vagus nerves, are not producing any significant stimulation. So in conclusion, we believe that it is GLP-1 derived from luminal microbiota that is signaling to crossing the colonic barrier, signaling to submucosal neurons and vagal nerve afferents, and possibly the underlying mechanism in this microbiome gut-brain axis, and suggesting to us that it could be the L cells that are potentially signaling this luminal microbiota. So I'd just like to thank Dr. Derval O'Malley and Maria Buckley for their help throughout the project, and also Chagas for providing the probiotics.